webinar. But before we start about it, let's pray 2 minutes Mahamrityunjay Jap, which we do before our webinar for the well-being of all. So let's have 2 minutes of Mahamrityunjay Japa.
बंधना मृत्योर्मुक्षि यमामृता यजामहे सुगन्धि पुष्टि वर्धनम् उर्वारुकमिव बन्धना मृत्योर्मुक्षि त्र्यम्बकं यजामहे सुगन्धिं पुष्टिवर्धनम् उर्वारुकमिव बन्धनात् मृत्योर्मुक्षीयमामृतात् त्र्यम्बकं यजामहे सुगन्धिं पुष्टिवर्धनम् उर्वारुकमिव बन्धनात् मृत्योर्मुक्षीयमामृतात्.
So friends, we are here today, yet again, 12th webinar of Friday 3D webinar series of dilemma, debates, and discussions. Me and Amit, we enjoyed this series of 3D webinar tremendously. Every Friday, 8 p.m., we all have made till now. And today will be the last webinar. We all started keeping in minds all the gynecologists of Maharashtra who wishes to improve their infertility practice. So our programs were not necessarily focused on ART, but they were mainly optimizing infertility practice in your day-to-day life. Apart from ART, it was more on IUI and other factors. Many young team members, they have taken care in the initial 6 to 7 webinars, and then our vibrant managing committee members, Dr. Varsha Vaste, Prerna, Manju Bog, and now Sadhana Patwardhan, our income chairperson. I think Sadhana, I can't calculate, maybe 8 years down the line, you will be the chairperson of MSR, I'm sure. What me and Amit have enjoyed with these, we had tremendous educational learning, 8 p.m. to 9:30, making those programs, getting those national, international people for the keynote on different themes. He was getting many SMSs, WhatsApp messages, and mails, which topics, because for the public demand, we repeated polycystic ovaries with the different set. We all know that polycystic ovary is a large topic and you can't cover it even in an entire day. So friends, this all would not have been possible just by me, Amit, and a fantastic team and the faculty and the delegate. There is a tremendous academic contribution by Zydus, the GM, not doctor, always I say you doctor, no? Sudeep, ek din Dr. Bani Jatu. Sudeep Mukherjee, our academic partner. I don't know whether we should call academic partner or whether we should call him MSR partner or All India partner, because he is doing academics across the country with many organizations. Thank you, Sudeep. And today, Sadhana, I'm not going to miss your name because you are the one who helps us making all those cards, communication, along with on-for-us team and posting them on various groups. Thank you so much, Sadhana, and posting them on the website with the links, all the background work you do. I love you more and more. Now, most important thing is COVID has taught us many things in life. And one of the best thing is coming out with the webinars. And these webinars are only successful because we have a fantastic, fantabulous digital partner. I don't think anyone in the world can compete with Conference. I'm aware Makran, Dominic, Mandar, Sagar, his entire team has done 8 international endoscopy webinars with me, and amount of requisite which has come to Makran because of his quality. The way their digital platform is, the videos and everything runs so smoothly. So today, from bottom of my heart, I wish to thank you, entire conference team. Without you, it would not have been possible, or probably me and Amit would have said once in a month, that's enough. Let's not bore it all the people with every Friday. But thanks to you that you kept us alive. Friends, we are not going to stop anywhere. MSR is never a sleeping chapter. We are coming up on 20th of December. Can I have? Yeah. So I request all the delegates, because I know we have locking in or many people from outside India, from Himachal Pradesh, from east of India and across, the like-minded people who wants to enjoy than having Arnab fighting with his debates. They love the debates of the academician. Please block this date. We are coming out with one of the best scientific program. It will be ART, infertility for a gynecologist as well as embryologist. In 2 parallel halls, we will be running this. So please block the date before Christmas so that you can enjoy Christmas till the year end. Thank you so much. And I request Amit, if you wish to say a few words.
Thank you very much, Sunita. And I endorse all what you have said. And I'm looking forward to seeing all of you and more on the 20th of December for our 1 day. I won't call it our annual conference, but maybe one of our last conferences before we hand over to the succeeding team. So I think without wasting much of time, I would like to hand over the proceedings to Sadhana to take over from here on. Thank you.
Sadhana, you can unmute yourself.
Sadhana, unmute.
Sadhana, you're there?
I think she's there very much.
Am I audible now?
Yes.
Yes, you are
Yes.
audible. Go on.
Yeah. So good evening, everybody. I welcome all the esteemed faculty of today, as well as my illustrious chairpersons for this webinar on genetics of folliculogenesis and ovarian failure. The basic aim behind today's webinar was a lot of the ART specialists are facing young patients with premature ovarian insufficiency, and it is a distressing situation, to treat these patients. We all should try to understand what goes behind in genetics of folliculogenesis and this ovarian failure. So we have eminent faculties today. They'll be dealing with the etiology of premature ovarian insufficiency, the genetic markers for these ART, and the investigations which we should be doing into the premature ovarian insufficiency patients. Without wasting much of time, I would call upon my first chairperson, Dr. Manjeet Mehta, to chair the first session, in which Dr. Anuba Shambhu, who is from the Pasteur Institute, France, who is an experienced geneticist. Dr. Manjeet Mehta, I would like to introduce to the audience. She is a consultant reproductive geneticist from Mumbai. She is a doctorate in human genetics from Jaslok Hospital. She is a postdoctoral fellow from North York Hospital, Toronto, and St. George's Hospital, London. She is the director of Genetic World, Mumbai, and genetic clinics in Bombay, Ludhiana, Varanasi, and Delhi. Not only this, Dr. Manjeet Mehta is also the managing committee member and national member of National ESAR, and she has lot of research projects, contributed book chapters, and has lot of publications in many of the journals, national and internationals, and she's an invited speaker for national and international conferences. So over to you, Dr. Manjeet.
Good evening, everybody. Thank you, first of all, for the kind introduction. I'm grateful to MSR for giving me this opportunity. And it's been a very interesting series that we've had so far. And ending the series with a super exciting topic that we're having today. Let us go to the first speaker, and the first speaker is Dr. Anu Bashambu. Dr. Anu Bashambu has a background in clinical genetics and stem cell biology. She is a senior group leader at the Pasteur Institute in Paris, France. And in their team, they are working on understanding the mechanisms that govern cell fate choice during the human gonad development. This is a very interesting topic, and we would be getting more information on this topic from her during her talk today. For this, she uses various technologies like next generation sequencing and in vitro tools. And using them, they try to define a novel genetic and epigenetic anomalies and pathways that are associated with atypical development and function of the testes. A lot of work has been done by her group on DSD, and she's got several publications, over 90 peer-reviewed papers in esteemed journals like NEJM, American Journal of Human Genetics, JSM. So without coming between you and the talk, let me invite Dr. Anu to please start her presentation.
Thank you, Manjit, for such a kind introduction. And I would like to thank MSR and Deepak and Dr. Patwardhan for inviting me. Unfortunately, I would not be talking about DSD today, but we can discuss it if anyone is interested. I'll share my screen here. So let's see. Can everyone see the screen in the presentation?
First, open your presentation, madam. First, open your presentation.
I have opened it.
Yeah, now click on Zoom app.
Okay. I'm using Zoom through the, so I'm just using application window, Microsoft, and I'm sharing it here.
Now click and share.
Can you share it from your side? It doesn't seem to be working here.
Mandar, can you share the screen from your side?
I think
Just a minute,
Deepak
sir.
also
Just a
downloaded
minute.
it, no?
Anu, can I do it for you? Is that fine?
Yes, please. Yes, please.
I'll do that.
Perfect.
Oh, cool.
Okay. And the thing is, you'd have, how am I going to move it from here? That's the problem if you share it. That becomes a bit of an
You can
issue.
say next. You can say next slide to him.
Saying next.
Is that comfortable?
Okay. It's going to be a bit of a thing. Anyway, so let's see. So yeah, I'm going to talk about the genetics of POI. And next, please. Well, I don't think I need to explain to you all what POI is, but technically it is arrest of menstruation before technically 40 years of age. The incidence is very variable. It is presumed to be about 1% before the age of 40 years and even lesser before the age of 30 years. The etiology of POI is rarely varied. It includes, of course, cytogenetic and genetic causes, but at the same time, there could be causes that are infectious, iatrogenic, autoimmune, and metabolic. Now, lots of isolated POI cases, they appear that they are sporadic, but if you look at that, about 10 to 15% of them have got affected first-degree relative. So it shows that genetic has a significant contribution to the etiology of POI. And when I say genetic contribution and genetic etiology, and it's not only POI, and I'll emphasize it throughout my talk, there are other disorders of reproductive system, including DSDs and male and female infertility that may form different regions of the same spectrum. And I hope I'm able to convince you by the end of this talk. Next, please. So here, at least in the Western countries, the typical strategy for the diagnosis of the patient has an emphasis on the importance of genetic etiology and genetic contribution to the cases of POI. When a patient presents with the suspected POI, of course, it undergoes the FSH and estradiol screening. It undergoes the chromosomal analysis and FMR permutation analysis, which is a major cause of POI. However, if the patient is still idiopathic, then depending on it is syndromic or non-syndromic, there is a whole range of genetic tests done, which could be by candidate gene screening, depending on the presentation. It could be a panel screening, it could be whole exome, and in extreme cases, whole genome sequencing. And then depending on what the results are, a multidisciplinary medical team takes over. That includes the endocrinologist, the gynecologist, geneticist, counselor, neurologist, oncologist, etc. Next, please. So as I said, it has a varied etiology, and each of the causes have a different frequency that it can contribute to POI. Most obvious are the aneuploidies, X chromosome defects, and FMR permutations, as well as single gene defects on the X chromosome. POI might present, as I said, as a part of a syndrome that include BPES most importantly, or premature aging syndromes like Bloom's, Werner's, or Cappo. The isolated POI seems to have known genetic causes that have been verified and contribute to around 1% of all the cases of POI. These are the single gene variants. Next, please. And these gene variants can impact any part of ovarian development. As you can see, they could be the genes that are required. Well, I can't move my cursor here, but to the left of your screen, you would see the genes that are required for ovarian formation or gonad formation, and the genes that are required for follicular development. Even for follicular development, the genes required for every stage of physiology could be mutated, and their mutation can result in ovarian insufficiency. Even during folliculogenesis, next please. If you see in the upper panel, the genes that are involved in the maturation of the follicle could be a cause of POI, or the genes that are involved in meiosis, or that genes that are involved in DNA repair, the mutations in those can also give POI. I'm not going to sit here and give you a list of genes and say, okay, this gene does this and that's how it causes POI. You can get it in any decent review. If you want, I can give you a list of reviews. What I want to show you here is how do you identify a gene and say that, yes, this is a genetic cause of POI. So I'll start with some of the examples that we have from our lab. Next, please. The first being the gene NR5A1. It's a often nuclear receptor. Most of you would know it as a major cause of XY-DSD. And while we were working on it, us and other groups have shown that the mutations in NR5A1 throughout the length of the protein can majorly contribute to different forms of DSDs, with or without adrenal insufficiency. Now, NR5A1 is a major master stereogenic regulator. It's the mother and father of all stereogenic regulators, basically. Anywhere in the body where stereogenesis is taking place, NR5A1 is a part of that pathway. So it can give rise to different DSD, XY DSD conditions, and as well as male infertility. And the general consensus at that time was, next please, that NR5A1 mutations will affect the gonad only in XY chromosome complement and not XX. The reason being this paper that was published where they had a homozygous NR5A1 mutation in NR5A1, and this patient was a prepubertal girl that presented only with adrenal insufficiency, and the gonad was absolutely normal in terms of structure and function. So, next please. When we were analyzing our cohort of DSD patients, we identified these 4 completely novel mutations in 4 families in patients that presented with different degrees of XY DSD. However, next please. When we looked very carefully at the pedigrees, we found that at least one other individual in the family whose XX was carrying the same mutation and showed some degree of ovarian insufficiency. So the blue, pardon me for being stereotypical, blue is the XY and pink is the XX, but the blue are the index cases. And if you look at family 1 and family 4, which are the top and bottom right of your screen, there the mother had secondary amenorrhea, the premature ovarian failure, where they gave birth to a child and that had depletion of ovarian reserve over time. Then if you look at family 2, the girl who's the sister of one of the index patients, had primary amenorrhea. In family 3, the sister of XY individual had secondary amenorrhea. So it was a whole range of ovarian insufficiency that we saw. And all these patients carried exactly the same mutation. We had at that time 25 isolated cases in the lab because, as I said, POI is not our major pathology that we work on. But when we screened those isolated cases, we found that there were 2 other individuals that carried mutations in SF1 or NR5A1. One of them showed a kind of ovarian dysgenesis where there was only one gonad. It was fibrous with no follicles, whereas the other one, we didn't have the gonadal structure, but showed hypertrophic clitoris, suggesting that it must have been exposed to testosterone at some point of time in utero. Next, please. So all these mutations, they fell in very conserved regions of NR5A1. And with a lot of in vitro assays, we could show that they were affecting the biological activity of the protein. Next, please. So what we have shown you so far, that the mutations are segregating with the phenotype. The mutated protein is in conserved amino acids, and the mutated protein affects the biological activity of the protein. Now to the holy grail of genetic screening would be, we analyzed 4,000 alleles which were ancestry matched to these patients to see if the mutation was present only in the affected individuals or it had a contribution to the healthy individuals across the populations as well. And we did not find any of these mutations in 2,000 cases or 4,000 alleles. Next, please. So after doing all that, we could prove that yes, NR5A1 mutations are indeed associated with ovarian insufficiency. Because the numbers we did in the initial paper were quite small, so our number were about 4 or 5%. But as we did more analysis, next please, and other groups analyzed it, we know that now that the mutations in NR5A1 are responsible for around 1 to 1.5% of cases of POI. With varied presentation. And the incidence varies with the population. Next, please. And as I said, that we are finding a common genetic etiology on the cases of POI and DSD, suggesting they're a part of the same spectrum. So NR5A1 that causes DSD causes male infertility, causes XY DSD, male infertility, also causes POI. And then there are several papers coming which are showing that the same pedigrees, if you look at it, look at the pedigrees, there may not be a second patient in the family with POI, but they may have other reproductive anomalies. However, all of them are carrying the same mutation. Next, please. So I have shown you that there are mutations along the NR5A1 that can cause POI. However, I'm going to complicate it further. If you have one particular mutation, you can have mutations throughout NR5A1, and it will give you infertility, XYDSD, POI. But if you have R92W, if the arginine residue 92 is mutated, and you have XX chromosomal complement, you will have testes or ovotestes. Next, please. So basically, it started with the two sisters that were 46 XX males or with ambiguous genitalia, and they had this mutation in NR5A1. Since then, we saw 6 different families where this same mutation occurred, and then there are other groups who have reported exactly the same mutation that are associated with XX maleness. It is important. Next, please. Next, please. Okay. So I'll just come to that. For the moment, I have talked about the inactivating mutations of NR5A1 that can contribute to anomalies of reproductive function and reproductive development. POI. What about overactivity of NR5A1? It has been associated to endometriosis, which could be a cause of ovarian insufficiency. A particular mutation has been reported in a hyperandrogenic hirsute women that also has ovarian insufficiency, not classic, but the ovarian reserve was quite low. Next, please. So basically, the mutations in NR5A1 are associated with XYDSD with or without adrenal failure, a spectrum of phenotypes in XYDSD, XY spermatogenic failure, XX adrenal failure, XX POI, XX TDSD, OTDSD, or you could carry the mutation and be completely healthy. Why is this important? A patient comes to you for IVF. She has got POI because of a mutation in NR5A1. She can pass it on to the child that she conceives by IVF, and the child has a possibility to have either of these phenotypes. So it's very important to know what is the genetic cause in that patient so you can counsel the patient accordingly. Next, please. Okay. Again, coming to the same topic of POI being on the same spectrum as other reproductive anomalies, I'll give you an example of another gene, SOX8, which is a part of SOXE family. If you know, SOX9 is a major regulator of maleness in humans. SOX8 is kind of a brother of SOX9. It's more or less the same. And we show that if we have rearrangements of SOX8 or a mutation in a functional domain of SOX8, which is the HMG box, it can alter the function of the protein and cause XYDSD. So far, so good. Next, please. However, if you have mutations in other regions of the protein which are not as deleterious as the HMG box, the phenotype that you see is infertility. It's either azoospermia or oligozoospermia in XY individuals, or it is POI, again with varied presentation in the females. And the incidence of these mutations is even higher. It's about 5% cases of female infertility will have mutations in SOX8. However, the mutations in SOX8 are also present, or the variants in SOX8 are also present in the general population. The same minor at the frequency of about 0.5%. So you have to be very careful while interpreting that data. Nevertheless, it again reiterates that all the reproductive disorders at the level of development may have a same genetic etiology. Next, please. STAT3, which you know is a gene that's required for meiotic repair, and it is the known cause of POI. And it has been verified as a cause of POI. Recently, we've shown with our collaborators that it again can contribute to male infertility as well. So again, expanding the list of genes that basically gives the proof. Next, please. So I'm not going to go, as I said, in the list of genes. There are so many candidates, and they're increasing every day because of the advent of NGS, and everyone is using it. But there is only a handful of genes that have been unequivocally proven to be causative by robust functional assays, by doing complete case control studies, or by functional validations, or by animal models. You may have a sporadic case with a variant in a sporadic case of POI with a variant that you have identified by NGS, and you try and publish it and say, okay, this may be the cause of POI because I found it in my patient. But if it does not have a robust functional study, it does not have a robust genetic study behind it, it is very difficult to interpret it and give proper counseling to your patients. Again, I'm reiterating the emphasis on the case control as well, that your controls should be matched for ethnicity, for your whatever you may call the caste, the clan, the group, the region, the geographical location they come from, because there could be population specific variants. And we know that from the case of NR5A and other genes that there are population specific variants that are not pathogenic at all. So, for example, a gene in DSD called MAP3K1, which is very important in DSD, there is a variant that is published as pathogenic, but that variant, if you look at the South Asian Bangladeshi population, 30% of individuals carry that variant, whereas in a Caucasian population, that variant is not that common. So they did not look at the appropriate control population. Okay. We always say that the gene, if it is it's affecting the ovary, is expressed only in the ovary. That's not the case. It could be ubiquitously expressed. The genes that I gave you are the examples, but it must be expressed in the ovary. It must be expressed in the ovary, but not limited only to the ovary, because it can have a far range effect. So let's say any gene that is affecting the hypothalamic reproductive gonadal axis can have an effect on the development and function of the ovaries, and therefore cause ovarian insufficiency. As I said, the genes don't work in vacuum. So a gene by itself may not be causing the phenotype, but it may be affecting the downstream target, which by itself is not mutated, but is not properly activated due to an activation in the due to a mutation in the upstream gene, and that can cause the phenotype, whatever phenotype you're looking at. And it is important to know the genetic etiology to understand the pathophysiology, obviously. And as I said about in the case of NR5A1, and it is the case in many other genes, to provide counseling and fertility guidance. To predict the age of menopause, I'm not even going into that topic. It's a whole range of literature correlating the age of menarche and the age of menopause, and how that could contribute to ovarian insufficiency. But we can discuss it if anyone is interested. And if there are mutations that are predisposing you to the loss of ovarian reserve over time, then the possibility of cryopreservation of the oocytes or OTCs. Next, please. Now, with the advent of NGS, as I said, that there is a huge number of genes that are coming every day, and not all these technologies are relevant for diagnosis. For example, CGH or array panels or exome sequencing, you could use it for diagnostic or clinical use. However, they are not useful. Genome sequencing is not useful for screening or clinic. Even in research, it is very complicated to work on genome sequencing. What it does is it gives a range of variants that we have no way to verify, or or groups that just publish it without verifying that it actually is a causative gene or it's a causative variant, and that confuses the clinician a lot. How are they going to counsel their patient? Is their diagnosis correct? Because it has been published. But they don't see a phenotype that's similar to what is published. The reason is that probably is not the gene causing the phenotype because, next please. As I said, you need a huge body of work to establish causality before a gene is called that this is the genetic cause of a phenotype, including POI or any other phenotype that you're talking about. So first is the genetic evidence, which is the mutation or the gene variants segregating with the affected population? Second, in case of familial cases, it's rather easy if it is segregating with the affected cases and not there in the SIBs that are not affected or other family members that are not affected. If it is de novo, it's not always true that it is going to be causative, but it's more likely that it might be causative if the mutation is de novo. Again, I'm emphasizing, is the mutation absent from the appropriate ancestry matched controls? In a country like India, it is absolutely imperative where we have so many different populations in terms of population movement and population ancestries. Once the genetic evidence is strong enough, then we have to build up the biological evidence. First of all, that the mutation fits or the gene, it fits in the biological context. As I said, the gene that can possibly cause POI must be expressed in the ovary. It could be expressed anywhere else as well. But if it is not expressed in the ovary at the right time, at the right place, it is not going to affect the function of the ovary. End of the story.
Doctor, you have 5 minutes left.
Okay. Then a range of assays to predict the effect of the mutation on the protein, a range of assays to show that the mutation is affecting the protein, animal models, and the human organoid models, or the iPSC models, or the cellular models. Next, please. Okay. So I have been going on like, okay, you've got genes, you've got genetic causes of POI, you can identify new causes. How can you do? The question you should be asking me, what is it going to do for me? Even if I know this gene is causing the phenotype in my patient, how is it going to affect the patient care? Or how is it going to improve or not improve the IVF or ART if the patient is going to present for that? I think Deepak is going to go more into detail of these, but I can just give you certain examples. If you're giving gonadotropins to stimulate the intra-ovarian IGFI, because it will increase the effect of the FCH on folliculogenesis and increase the ovarian reserve. However, it may not give a proper response. The patient might be a poor responder, not because there was something wrong with the technique, but the patient is carrying a mutation either in LH or FSH or FSHR. That is not letting it metabolize that or use the gonadotropins that you are giving. That's not letting it reach the follicles and do its function. Glucosteroids along with GnRH and gonadotropins has been suggested as a good therapy for inducing ovulation or increasing the ovarian reserve and achieve pregnancies in poor responders. But they are not going to work if you have mutations in FSHR, NR5, or BRCA1 and 2, which are required for the function of these genes. If you have progressive loss of ovarian reserve and you have a mutation in a gene that you know is going to cause a progressive loss in ovarian reserve, for example, in cases of Turner's or NR5A1 and STAG3, you can advise your patient for cryopreservation. These are the uses of genetics for you.
Sorry to interrupt, madam. Your time is over. Your time is over.
Okay. So I can just stop here then.
Anu, you can conclude.
Yeah, just wind up your summarize, the take-home message or something.
So basically, my take-home message is what I have on this slide, that you need genetics to try and understand how your patient is going to respond to the treatment that you are going to do to them in terms to increase their ovarian reserve. You need to understand the genetics of the patient to give them counseling and advice before they enter IVF, and to give them counseling what are the possible effects that the prospective child can have if it is inheriting the same mutation that the mother is passing on to the child. Next, please. I think that is my last slide. Yeah, that was my last slide. So yeah, I mean, I just thank all my lab members, collaborators, clinical, scientific, funding, everything. And thank you, everyone.
That was a beautifully carried out talk. I mean, we just wish it would have gone on and on, because it is such a complex and such an in-depth topic, you know. I mean, you cannot do justice to it in such a short time. Anyway, for the in interest of time for the other speakers, we have a few questions coming in, but we'll be taking all the questions at the end. So thank you very much, Dr. Anu, for a very clear and lucid presentation. And thank you once again to MSR, Dr. Sunita, Dr. Amit, for giving me this opportunity. And this is the end of the first talk.
Thank you.
Sadhana, you have to unmute yourself.
Padmarekha, you can take over, please.
Okay.
We are already delayed by 20 minutes.
We are starting with the second talk, for which I'll invite Dr. Deepak Modi. We all know very well that he is scientist and head of molecular and cellular biology laboratory of NIRRH Mumbai, and he's very much part of all our scientific endeavors and a good friend of MSR too. So I now invite Dr. Modi to deliver his talk, take us further into the various aspects of genetics of premature ovarian insufficiency.
Thank you, Dr. Padmarekha. I'm audible. I suppose I'm audible.
Yeah, you are audible. Yeah.
Thank you. Thank you, Dr. Sunita, Dr. Patri, and Dr. Sadhana for having me here. And it's always a pleasure to talk at MSR. And I'm really glad to see that MSR really gets hold of all basic scientists all around and makes them talk to clinicians and make us talk to clinicians and clinicians talk to us, so that there is a free exchange of understanding of how the basic science translates to clinics. And that's what I'm going to do is I'm going to take over from Anu's talk, which I'm sure everybody enjoyed, where she really gave us a deep insight into the premature ovarian failure. But when we talk about the clinics, I'm going to transport you from the laboratory to the clinic for some time. And when we have the clinic, you would have a woman who, the first question you would ask is, after a series of investigation, is it okay, now I'm taking her for IVF, will she have enough follicles? If I stimulate her enough, how much should I stimulate her with? Will she ovulate? Will she respond sufficiently? What if she doesn't respond? Do I need to jack up more hormones? And so on and so forth. And these are questions which runs in every clinician's mind, I'm sure of that. And once you do the ovarian stimulation, most of you would land up getting the normal response, about 6 to 10 eggs, and you're happy. But the moment you start seeing more eggs being developed in the ovary, and you say that, oh, freak, this looks to be a high responder, and you would kind of reduce the gonadotropins to get here. But these low responders, I'm sure, are a nightmare for everyone, because she would produce less than 3 eggs, and then most of them would not have good fertilization. There's a high range of aneuploidy there. And once there are not enough eggs, in most of the cases, the cycle is cancelled. And this is where the whole dilemma begins. So this is not a spectrum where you are seeing, okay, she's completed, I mean, she's into menopause, but these are women who are responding lowly with the hormones. And then why should this premature ovarian insufficiency or premature poor ovarian reserve occur? So there could be two possible mechanisms. One is that, as Anu said, that there would be absolutely no or very few follicles in her ovary, and these are letting her ovulate a bit slowly. Maybe she's irregular cycles, or she's doing it a little slowly. And over the period of time, this follicles deplete, and she'll be presenting as primary ovarian insufficiency, or POI, which Anu has really dealt in great. But there could be dysfunctional follicles where they are not able to respond to gonadotropins, or they cannot generate enough gonadotropins. And these are the two possible reasons. And irrespective of these reasons, a woman will present in the clinic as most likely a woman who is infertile, and she has not responded to gonadotropins. So of course, when we talk about, so first thing is, as Anu said, you need to rule out whether she has enough follicle numbers in her ovaries. And for that, you would do chromosome aneuploidies and look at a range of gene mutations, which she has very nicely alluded through the new genes which have been discovered. And I'm sure Prochi will cover the classical genes which are used currently in laboratory for diagnosis of premature ovarian insufficiency. And my interest in this was long back in my PhD, where we could show that how in women with Turner syndrome in utero during the course of the normal ovarian development, most of these eggs die by the process of apoptosis. And by the time the baby is born, there are already very few follicles in their ovaries. And by the time she comes to puberty and she starts ovulating, very few women in fact ovulate, and they ovulate for a few cycles and then they get into ovarian insufficiency. And we could show that in these women, the follicles or the eggs have actually died when they were in utero. And large numbers of candidate genes have been identified, like Anu has said, NR5A1, and two of them very nicely she's explained, and there's several of them which are identified throughout the genome, which are the causes of POI. And there are also these X-linked variants on the X chromosomes, which could be leading to premature ovarian insufficiency. And this is this long list of genes which is ever increasing, and they could be classified as transcription factors, DNA repair gene alterations, or they could be genes involved in germ cell development, they could be involved in hormone pathways, or they could be also involved in immune pathways. And these could be one of the major causes of premature ovarian insufficiency. But what about poor ovarian reserve? So you did all this karyotyping. Hopefully you have been able to also look at several genes, and you kind of are now not sure whether she is premature ovarian insufficiency or she is having a inherently poor response. So what would you do in these cases is what I'm going to talk about in the next few slides. Basically, you guys are more well versed with the Bologna criteria, which says that a poor ovarian response can be defined as a previous POR less than 3 oocytes by conventional ovarian stimulation protocol, with a risk where the advanced maternal age is a risk factor, and they may have an abnormal ovarian reserve test where the antral follicle count is less than 5 and the serum anti-Mullerian hormone levels are 0.5 to 1. But now the Bologna criteria is slowly being replaced by the Poseidon criteria, where you have where you can group them based on AFC and the AMH count as well as the age. And here you have these women who are good risk, who are supposed to be young, or they have good amounts of AMH that is higher than 1.1, and you could have patients who have AFC less than 5 and the AMH is lower than 1.1. And these are the patients with probably premature or poor ovarian reserve. Inherently, the reserve may be low, but these are the ones who have insufficient ovarian reserve and may have sufficient ovarian reserve, but may have poor prognosis because the AMH levels are good enough. They're not PCOS, so their AMH levels are good enough, but still they do not respond. And that is this group of patients which are possibly having follicles in their ovaries, but the follicles just don't seem to respond to the gonadotropins. So administration of an inappropriate low-dose gonadotropin may lead to growth of low number of follicles in an iatrogenic poor or suboptimal response. By this, it means that when you started with the gonadotropin dose, the dose was itself insufficient, and therefore insufficient number of follicles developed, which gave you fewer eggs. So then in the next cycle, you'll end up jacking in more hormones with the hope, for keeping fingers crossed, that okay, this woman will now give me more follicles. But that often does not happen. And it is important that even before you starting on this hormone therapies, why should some women have such poor reserve, and can we identify them well in advance? So suppose if, let's say that there are these poor responders. Number one, there could be insufficient hormone production. The hormone itself may not be of good quality. That is, they have some issues with the hormone shape, the hormone structure, or there could be insufficient receptors of these hormones, or inefficient receptors. Now, in a typical IVF setup, you're going to give this FSH and LH exogenously, which are recombinant, very well characterized, and very nicely tested. So this does not seem to be the major rate limiting factor in a poor ovarian response, but could there be insufficient receptors or inefficient receptors? And this is what we are going to check using genetics. And genetics comes into picture in these conditions where the receptors are involved. So basically, when we look at premature ovarian failure, in poor ovarian response, you could have issues with the AMH or the AMH receptors, or you could have issues with the FSH or FSH receptors. And we also use these hormone levels of AMH, FSH, and estradiol for making a judgment whether this woman is responding appropriately or not. And AMH, you are well aware that anything 0.5 to 1.1 is an indicator of a POR. Antral follicle count less than 6. There's always controversies on these levels. The estradiol less than 80 picograms per ml or FSH greater than 10 milliIU per ml are diagnostics for POR. And I'm sure you all have seen this diagram that there's a very nice correlation between FSH levels, AMH levels, and AFC. But however, we know that antral follicle count has a lot of issues by themselves. We are still not sure whether 5 or 6 is the cutoff, and there's a lot of inter-observer variation. AMH has been something which is we all swear by today, and low AMH or very, very low AMH are indicators of POI or POR. However, AMH also is very, very tricky. Its interpretations are not as easy as it sounds on paper. We know that AMH values correlate very well with antral follicle count only for a few years, between 25 to 35 years of age. After 30 years of age, the AMH and the antral follicle count just do not seem to match for some time, and then they match at the end of near menopause. Now, also we know that the levels are age dependent. Often you have to land up normalizing them. They are hard to interpret, expensive, and you know that you cannot apply them to women with PCOS or where AMH levels inherently are abnormal for some other reasons. So genetics comes in very, very handy because these are not dependent on certain values. These are like yes, no kind of a test. You can say, okay, there's a polymorphism, or there is not a polymorphism. You can say that there is and it is irrespective whatever time of the cycle you do, whatever time you don't have to really wait for a particular day of cycle and so on and so forth. And to explain you to the audience who are not very well versed in molecular biology, I'm sorry, it is going to be a very basic repetition of people who are not or who are very well versed in genetics, but I love to explain the concept of polymorphisms like this, that I spell my name as DEEPAK. Now, if it becomes DIPAK, it doesn't really matter much. Overall, it's not going to change much, but when it comes to legal consequences, a DIPAK spelling would be completely problematic over a DEEPAK. And what we try to look for polymorphisms, we are trying to look for these minute changes here. What Anu spoke about is a missing K completely. So instead of a Deepak, it becomes Deepa, and my gender changes, and that's a very strong change, which would obviously give you a much stronger phenotype. But these are very small changes which don't give you a drastic phenotype, but they matter in certain circumstances. And as of today, such polymorphisms have been identified in the FSH receptor gene, the LH receptor gene, AMH and AMH receptors. I'm going to share some data on them. So FSH receptors and its polymorphisms amount to improper amounts of FSH receptors, or there could be improper binding of FSH to FSHR, or there could be improper AMH or AMHR, and they can create a problem. So we know that our nucleus is all these ATGCs, and if we are able to count all the ATGCs from a single cell, we would actually fill up a huge phone book. The younger generation may not have seen it, but we all know that there are these huge phone books which are lying in our house, and we would hunt through the phone numbers, and that we have to literally go through this entire genome to find what are these changes. And each of these changes can be divided into genes, and these genes have specific functions. Now, each gene has a promoter and an open reading frame. Now, if there are mutations or polymorphisms in the promoter, the amount of the protein which is made out of it is insufficient, and that can lead to inefficient production. Or if there are genes or polymorphisms in the open reading frame, then it could lead to improper function. The protein may be produced in adequate quantities, but the functions may be heavily compromised. And when we say functions may be heavily compromised, look at this here, that a single nucleotide change in the DNA would translate to a very little change in the shape of the protein. And such a shape of the protein may not have a dramatic effect, but when the hormone wants to lock into its receptor, a slight change in the shape may not allow adequate locking and not enough signaling to occur. So you may give jacks and jacks of exogenous hormones, but because the hormone cannot bind efficiently or cannot process the signal efficiently downstream, the follicles will refuse to respond. So there are polymorphisms, I said, in FSH, FSHR, AMH, and AMHR genes. And here is one such example, which was published from our lab several years ago, where we could show that one of the polymorphisms, which we call it at position 380 in the FSHR gene, where the change is from a T to A. So if your patient has double T, or if your patient has double A, let's take for example, the basal FSH levels will not change at all. But look at the amount of ampules of FSH required. Those who have double T will require more amount of FSH as opposed to those who have double A. And if you inject same amounts of FSH, the double Ts will respond poorly, while the double As will respond at a very, very high amounts. So you can look at the difference, a single nucleotide change, not really doing anything to the normal physiology, not depleting the ovarian pool, but same amount of follicles, but they don't like to see the hormones. And with one change in nucleotide, you can see a differential ovarian response. This is one of my favorite slides. This is from Dr. Mahale's lab, which they published in 2015 in JCEM, and where they could show that there is another FSH receptor polymorphism in the promoter. So here we could show that this minus 29 GA-FSHR polymorphism, when there is a GG nucleotide, you can see beautiful FSH, the green color here, the receptors is nicely seen on the granulosa cells. But if it is a GA, you don't make much of a difference. One is coming from mother, one is coming from father. But if your mother and father has given AA at that position, look at this, absolutely very little amount of FSH receptor coming onto the cell surface. So that means this is a receptor mutate, this is a gene mutate or a polymorphism, which is not allowing enough receptor to be built up in the system. And look at this. Exogenous FSH, the women with AA require higher amounts of FSH. Still giving higher amounts of FSH will not produce enough number of pre-ovulatory follicles. The number of follicles retrieved would obviously be lower, and about 60% of the women who have an AA genotype will be a poor responder. So FSH receptor polymorphisms can strongly predict ovarian response to exogenous FSH. Similarly, we have a lot of data from Dr. Mahale's lab in our institute, where now Dr. Padma Rekha and Dr. Sadhana Desai are collaborators. And we could show here that with a certain polymorphism, you can have higher amounts of, with the CC polymorphism in the AMH receptor, you require higher amounts of exogenous FSH to induce same amount of response, and yet you may not get an adequate response, even still giving that quantity. And so if we look at the data, we could show that FSHR minus 29 has one of the strongest association, and AMH receptors and another polymorphism and AMH receptors also do the same. But remember that these are not very, very accurate tests. That means that not every patient with a POR is going to have these polymorphisms or mutations, because as Anu said, that they need not necessarily be in the FSH or the FSH receptor or the AMH genes. There could be hundreds of such changes, and many of them are not very well characterized. And remember that in this case, we have classified the patients as per Bologna criteria. And I'll be very honest that it's not even rightly the Bologna criteria. And we need to classify these patients based on Poseidon criteria as yet. But what is the way forward? What could be the way we could do that, kind of improve the efficiency of these hormones and the genetic testing, so that we could help you to predict that, okay, how many of your patients could have poor ovarian reserve at one go itself. So we took help with a team called as POVM in Bangalore. This is artificial. They deal with artificial intelligence, and they first looked at the data. And this is a lot of data, of course, not just from our lab, but from several parts of the world. And they could show that there's a very strong correlation. But whatever data is available as of today, the minus 29 polymorphism, remember I showed you the promoter polymorphism where they don't make enough FSH receptor, it is the strongest correlation with ovarian response. What they tell us now, that if you just use AMH and AFC, your positive predictive value is 86%. But if you now combine AMH, AFC, age, And the receptor polymorphisms, you will be correct 93% of times. So you can improve the efficiency of AMH and AFC as a diagnostic criteria if you pool in the data of the genetic polymorphisms, mainly the FSH receptor polymorphisms. And when we apply this algorithm to figure out what is a major contributor in the diagnosis of poor ovarian reserve, what they could tell us was that when you use BMI or age or AFC, AMH or basal LH, they all contribute in predicting whether this woman would have a good response. But when you apply -29 polymorphisms or a 307 polymorphism, don't bother about these numbers. What it tells us is that when you apply genetics, your ability to accurately predict a poor ovarian response improves dramatically. But even as of today, I would say that we have not reached 100%. We are somewhere around 90 to 95%. But this work is ongoing. I'll be happy to tell you that this company with whom we are closely working with will soon come up with an app where you can feed in the age of your patient, the BMI, the AFC count, AMH values, and also if you have the receptor polymorphism data, this dashboard will be effectively able to tell you that whether this patient will be a POR or she would respond normally. And if she's a POR, on day one itself, you can take a decision that you will need to give her higher hormones, or you can prepare her and the family that, okay, you might have a poor response and we should be prepared with additional cycles to collect the ovary, to collect the eggs. Now I'm going to switch gears here and make you take away from genetics. This is something very interesting. I landed up in a very different situation. My friend in IIT and one day just called me and said that, look, there's this a chemist here who is very much interested in determining how ovaries respond to FSH. And I said, well, how does a chemist come into picture? And I was expecting that she would tell me some structure of FSH receptors or so on and so forth. But she actually landed up giving some very interesting insights. And I'm going to share you the data. So she's Dr. Urmila Devekar. She's from University of Illinois at Chicago. And she also runs her own center called the Vishwamitra Research Institute. And these are some of the slides which she's given me to share with you all. And I'm really excited with this, that this is something quite interesting. So what she has developed is a very, very novel idea. So she works on particle crystallization, that how small particles crystallize together and form a bigger one. And the way she has developed a lot of mathematical formulas. So she's a theoretical chemist, and she looks at a lot of data and makes a lot of predictions. So she said that the way these particles grow, even the follicles must grow using the same mathematical equations. So she used a lot of mathematical equations and came up to a model to predict optimal hormone doses using principles of something that she calls optimal control therapy. Frankly, if you ask me how this math is worked out, I have no idea. But what she's shown me very wonderful data where she says that if you have the follicle diameter and the amount you have administered FSH for the first 2 days, she feeds into her algorithm. The algorithm undergoes a lot of iterations. And by the end of it, she would give you an optimal next set of doses that she would require to have an adequate ovarian response. So I said, okay, this is all theory, show it to me practically that it should work on some patients. And this is interesting. She has a very small data set as of today, but we can really see here that this was the red one here is the clinician devised dose, where the clinician either decided the dose based on the ovarian AFCs or so on and so forth. And the green ones are the ones which are decided by the computer. And you can see there's a huge difference in the amount of dose required to induce ovulation. And what did she get actually? So when the same patient was compared at one time when the clinician gave her, the clinician decided dose, and the second time in the same patient, the dose was titrated according to what the algorithm told her. And instead of 4, they got increase in number of oocytes with the lower amounts of FSH that was actually required. So she thinks that we are actually jacking in more of FSH most of the times, and you don't require increasing FSH with rather a staggered FSH. This is another one where she says that, okay, day 2, day 4, you continue giving the same FSH. Usually the clinician had increased the dose, but in this patient, the required optimal dose was far lower than what the patient actually required. And in this case, the number of follicles increased by by 120%. So this seems to be promising. And when we actually look at the mathematical calculations and the theory behind it, it makes sense that, how the particles come together and grow. Follicles also, as a biologist, I can tell you that the follicles come together and start growing in size in a very, very predicted fashion. And this is what coming from computer and chemistry, it's actually getting into biology, and clinics probably is going to make a change. So what she summarizes is that a mathematical procedure and a software has been developed, which based on the size and the distribution of the follicles per cycle and the function of drug dosage used, you can customize the amount of FSH required. And by this, she says that you can reduce the drug dosage by 20 to 40% and improve the test outcomes by 70 to 75%. And this is a huge achievement because both hormones are always problematic. We don't have enough hormones many a times, and we probably land up jacking too much extra than what is not required. So take-home message from my talk is that poor ovarian response can occur due to genetic factors where you already have low numbers of follicles at birth, which could be largely genetic, or they could very rapidly decline as the time progresses, where also genetic factors play a role. But they could be also because of failure to respond appropriately. And failure to respond appropriately has genetic basis in FSH and AMH receptors. And these could be reasonable markers to predict poor ovarian reserve. And if you use the genetic markers to predict poor ovarian reserve well in advance, you can optimize the patient therapy and you can give more personalized therapies to your patient rather than, giving an idea of one size fits all. And time is coming now that we are going to start using artificial intelligence to now not just decide how much dose to give, but what is going to be the fate of this patient throughout the course of treatment. I think that's the last part of my talk, and I acknowledge our director, Dr. Smita Male, who has been very generous enough to involve me in these projects, and Ramnath from SPOVAM, who has done a lot of analysis for us, and Dr. Urmila Devekar, who has really brought me this completely new perspective in predicting ovarian growth. And of course, all this requires money, and ICMR, DBT, and DSD are acknowledged. Thank you, and I look forward for more questions.
Unmute Padmarekha. Padmarekha, unmute yourself.
Yeah, thanks for that excellent genetic perspective on ovarian response, and also introducing us to the newer perspectives of artificial intelligence coming into our clinical practice. So we'll continue with the next talk now.
We move on to the third speaker of today's webinar, and I call upon Dr. Roya Rosati, the chairperson of this talk. Dr. Roya Rosati, over to you, please.
I think, Sadhana, can you move on yourself with the third speaker? I don't see Rosa here.
He was here, very much here, actually.
It doesn't matter. Just
Okay.
take the speaker on.
Can I have the CV slide of Dr. Prochi Madan? Yeah. Our next speaker is Dr. Prochi Madan, who is the honorary consultant geneticist at Jaslok Hospital and Research Center, Mumbai. She has started the genetics and PGD laboratory at Jaslok Hospital in 1999. She has introduced the FISH technique in India for PGD, prenatal and postnatal diagnosis, including leukemias in 2000. She is a senior genetic counselor on board of Genetic Counseling India. She has obtained Lady Tata Memorial Scholarship, the Young Scientist Award, the Dr. I.C. Verma Lifetime Achievement Award. Madam is also the part of Dr. Firuza Parikh's team, which has reported the first births in India after PGD by FISH for translocations and inversions. I welcome Dr. Prochi Madan for your talk.
Hello. Yeah, just a minute. Can you see my presentation?
Yes, but put it on screen, I mean, on slideshow.
Yeah.
Madam, go to the slideshow.
Yeah, just a minute. Is that okay? Have you got it?
Yeah, that's fine.
Just a minute. Yeah. So thank you for the introduction. And today I'll be talking on the relevance of genetic testing in primary.
Make it a slideshow, madam.
Pardon?
Make it slideshow.
Slideshow.
Yeah, click on slideshow button.
Okay.
Yeah, that's great.
Yeah. All right. We have heard of all the different gene mutations and all that. But now let us start again from a little earlier on, when there were not so much of genomics and gene studies, and there was cytogenetics, so the chromosome abnormalities. And so let us just give a brief overview of primary ovarian insufficiency. This was also sometimes called POF, or premature ovarian failure, and it is defined as the loss of ovarian function before the age of 40. It presents with ovarian dysgenesis, primary or secondary amenorrhea, idiopathic in most cases. There is increased FSH, decreased estradiol and AMH. The age of menopause is an inheritable trait. So POI has a strong genetic component. Generally, there are X-linked disorders in females with POI, and studying this and coming to know of whether it is present, whether any genetic mutation is present, is very relevant in today's world, because there is a trend for late pregnancies. And therefore, clinicians need to be aware of common genetic tests to check for genetic conditions which may affect the fetus, and then to do the appropriate testing to prevent this, because there is no cure for most genetic disorders. If they know it is a case of POI, they can start with oocyte cryopreservation earlier on. But though we say there is no cure for most genetic disorders, you all now have heard of the CRISPR and many other techniques. There is enzyme replacement therapy, so gene editing with CRISPR. And there is one more which we are doing in our IVF department with Dr. Firuza Parekh, and that is pre-implantation genetic testing, which was earlier called PGD, or pre-implantation genetic diagnosis, because this can help in preventing the conception of a child with a genetic abnormality, because we will be doing the genetic tests on the prior to conception on the trophectoderm biopsies of the blastocyst. There is one more recent success now, and ours is the second case in India, where we have been successful in doing in a patient with thalassemia. In order to cure this, you can have a bone marrow transplant from an identically HLA matched person, but getting this is very difficult. So another way is for the mother to do the IVF, even though she is fertile, get a large number of embryos, maybe with multiple cycles, and then send the biopsies of all of them for testing. The first thing it will be checked is to rule out the genetic disorder. So in this case, thalassemia, we only see that the child is not affected, homozygous. Normal or heterozygous is okay. Next, we check to see whether it is HLA matched, and very few will come as HLA matched. And then we have to see whether it is chromosomally normal also and not a Down syndrome or something. So all those tests are done, and there has been a success. The first case was reported by Dr. Manish Bankar in 2019, December, I think. And ours is the second case in India. So people have to be aware that this is now possible even in India, and that is how it goes. So now what are the etiological factors in ovarian failure? And Dr. Deepak Modi has been very kind to share some of his slides. So there can be, the major is abnormal karyotypes. So chromosome abnormalities, of course, most of them are idiopathic, but there are chromosome abnormalities, then there can be fragile X pre-mutations. And this is what I will be stressing on in today's talk. There can be antithyroid antibodies, anti-adrenal antibodies, anti-ovarian, and so on. And of course, the large number of cases are idiopathic. So let us move on. And well, I started my career in human genetics in 1973. In those days, there was no chromosome banding also. And we were just doing solid staining. And so I did my a PhD. So we had to basically check for numerical abnormalities. So my colleague took up Down syndrome and I took up disorders of sexual development, basically abnormal sex phenotypes. And so we found cases of Turner, Klinefelter's, ambiguous genitalia, and many others. And these few pictures are taken from those days. So as you all know, there's short stature, web neck, secondary sexual characters absent, and all that. You can see the shortened metatarsals and metacarpals. And so there can be either it's a single X chromosome, so that is 45X, but there can be deletion. So you can have a deletion of XP, or you can have a deletion of XQ, or there can be an isochromosome X where the XP is deleted and the Q is duplicated. And sometimes this can get more complicated also. There was a case very earlier on where the X chromosome was almost double the size, and maybe the isochromosome part was again being duplicated. So these are some of the things. Now, one thing to remember is that FISH, fluorescent in situ hybridization, is a very quick method of checking for these such numerical chromosome abnormalities here. But if there is a case of isochromosome XQ, you will not pick it up by FISH. So you are looking for Turner syndrome, but only if you see one signal for the X chromosome, then that will be picked up and you will say, yes, it is Turner. But in an isochromosome, because we have the probes for the centromere. So if you see two signals of the X chromosome, we will give a FISH report as normal, and therefore karyotyping is also very important. Of course, these days microarray is also there, which gives you much more in detail of the micro deletions and duplications, but microarray cannot check for any balanced rearrangements, balanced translocations, and others. So here you can see deletion of XQ and so on. So you can have even mosaicism. So what is mosaics is that there are some cells which are normal and some cells are abnormal. So you can have either pure 45X or XO and XX. And when there are normal cells, then the phenotype is not that severe. So in X chromosome mosaicism is therefore more common in women with secondary amenorrhea. You can also have X chromosome balanced translocations with the X chromosome and so on. So this was a case where there was an X chromosome translocation, very rare. So this part, the lower part of the X chromosome has gone to the upper part of chromosome 4. Now this actually we found in a child with developmental delay. And therefore we presume that this may not be a perfect balanced translocation, but there is a small micro deletion on 4P, and that could be having the girl's features. But probably her mother may be having this. And it is seen that because during meiosis, there may be a few rearrangements, and therefore the child may have a similar looking karyotype, but there may be a small micro deletion which is causing a problem in the child. So such things are known and you have to be careful about it. Now, mosaicism is known to vary the percentage in different tissues. And therefore, we have studied even the follicular fluid cells, blood, buccal, urine. And you can see by the fish the different patterns. And so even hair follicles can be studied. So X chromosome mosaicism is important because if you know that there is a maybe a Turner syndrome mosaic or someone, then ovaries can be cryopreserved if the diagnosis is made early enough. So phenotypic females with primary amenorrhea may occasionally show a karyotype of 46XY. So nobody is suspecting it, and suddenly you find XY, and then this would be androgen insensitivity syndrome, also called testicular feminization or female pseudohermaphroditism. And I got a number of these cases in my days of the PhD. And again, this can be complete or incomplete form. And prophylactic gonadectomy is advised around puberty because there's a risk of developing gonadoblastoma. So this was a married lady, very well developed, good breast development, but the karyotype was 46XY. And so this is called incomplete testicular feminization. In a complete form, the external genitalia totally normal. And this was a young child raised as a boy, but showed both one side and an ovotestis on the other side. So this is a case of true hermaphroditism. So these are all the disorders of sexual differentiation. Now technologies have evolved since the days of karyotyping and FISH. There is array CGH and SNP microarrays and PCR and next generation sequencing, and this has made the study of all those genes possible, which Dr. Deepak and Anu spoke about. So now we'll start again from the early days when these gene tests were not there, to detect fragile X by the karyotype method. And we started this, I started the department in Jaslok in 99, and by 2000, we had already standardized the process to check the fragile X. And you can see here on the X chromosome, a part which is broken and hanging. This is the fragile X chromosome. That is why it is called fragile X. And you have to be very careful because a chromosome number 6 will have the same size and shape. If you have not done banding, you may mistake it for chromosome 6. And therefore, this was very important to check for and to give a good result. So these are the children with fragile X, and basically they're very prominent ears and autistic features. And you all will be knowing that this is caused at this region, there are repeats. So trinucleotide repeats of CGG. And in a normal person, the repeats are up to 40. So it goes CGG, CGG, CGG up to 40 times. And that is normal. And up to 45. And when it goes from 45 to 54, then this is a gray zone. So because from 55 onwards up to 200 is the pre-mutation. So this in every generation, the number of repeats keep on expanding. And so a mother has to be a carrier of a pre-mutation, and her child, especially the male child, the repeats will expand to more than 200, and that child will have a typical fragile X syndrome. So this is why to know this and to get it tested in POI is very important. So it was also called the Martin-Bell syndrome, and the gene involved is FMR1. And the children are hyperactive, autistic. So this is what I've told you already of the repeats. So besides this, the male carriers, so there can be males who, now they have only one X chromosome, and those repeats may be in the pre-mutation range, and they don't manifest the fragile X, but they can get things like a ataxia syndrome as an adult. Similarly, females who are carriers, some of them will have premature ovarian failure and a low ovarian reserve. And therefore, in order to be able to exactly diagnose these cases, you need to study them. So the different associated disorders are, as I've told you, POF, POI, and the tremor ataxia syndrome. And very recently, they have again defined another one, which is Fragile X associated neuropsychiatric disorders. And you see that some of these women are very, very hyper anxious even during the pregnancy, sleep disorders, and irritable, and a lot of other things. And so you can suspect that I should check for the Fragile X carrier status. Recently, I got 2 females tested, and one is again a dilemma because her repeat size has come to 54 and the carrier starts from 55, but she is showing all these features. But again, I asked all the experts even abroad, and they said that from the sort of range of even around 60, it will not expand to a full mutation in 1 generation. So her child can be a carrier if that particular X chromosome has gone in. So she already has a child with autistic features. Tests done were normal, even the Fragile X. But then she was still very concerned, and therefore I told her that get your testing done because maybe your son has inherited your normal X chromosome. And this sometimes it just slips our mind, we may not think of it. And therefore these are very important. So, so the other female I tested, she was negative and she had the repeats in a normal range. So genetic counseling is very important, and the doctor should be aware of what tests to do, especially this. So World Fragile X Awareness Day is 22nd July, it comes in July. And even these days we say that test the oocyte donors, because sometimes the donor may not show any symptom and it will be totally, you will not suspect it also, but if you have not checked, then there have been cases where a normal donor was Fragile X positive. And therefore this is very important, and that is why I want to sort of, focus on it. This is sort of, it is called X-linked dominant with incomplete penetrance. So it passes on. So this is a carrier female, and her sons can be again carriers because they are not, their repeats are not beyond 200. And there's one affected male, and the carrier can have a daughter. All the daughters of carriers will be carriers because males have only 1 X chromosome, and that will go to the daughter. So all daughters of a carrier male will be carriers, and then they can have the Fragile X positive sons. So that is why this is very important. So now the expression of FMR1 in normal women, pre-mutation carriers, and therefore this is a slide again taken from Deepak, and just showing that how the expansion takes place.
Sorry to interrupt, doctor. You have 2 minutes left.
Okay, sure. So I was asked to give some Indian data, and so this is our Just Look data. And we have tested, of course, this data is not right updated, but quite recent. And so we had 958 cases, and we found, especially now focusing on the POF cases, we tested 102, and out of that 6% who were pre-mutation carriers, and 6 is not a low amount, and therefore you have to be very careful and aware of this, and definitely ask for the testing. You may be knowing that there is a Fragile X Society of India with Mrs. Shalini Kedia as the founder, and this is their website. And in their data, they have about 900 families. Some have 2 children, some have 3 children affected, total about 1,200 children in India in her database. There are going to be many more. And I took some data from Dr. B.K. Thelma also. They have done total of so many 200 cases, and they have found 98 pre-mutations. Of course, the pre-mutations, these are the total, not from the POI cases, because many people reporting PO, or not asking for the test. So about 20% of female carriers have POI worldwide, and among the India limited, there are 33% with POI. but many are not testing it, and therefore there are few cases, and there are some people earlier who have reported negative also. So you can do a prenatal diagnosis for Fragile X if the conception has already taken place, but so this also has to be informed to the couple, and genetic counseling is important. And if they can afford it, then they have to be given the option of pre-implantation genetic diagnosis. So all this I have already talked about, so we'll just go through this. And yeah. Now this was a chromosomal part, and there are different gene mutations with all the new tests, and that Dr. and Deepak have already spoken about. So these are some of the genes. And so that is the end of my presentation. Thank you.
Yeah. What is your taking home message? Is there any conclusion?
Yes.
Finally, as a clinician, what do you advise?
Yes, so I would say that even among the medical fraternity and even among the society, the awareness has to be important. And for this, sort of, once they are aware, and that is why I was stressing on the number of cases in India also, and it should be that this should become one of the standard testing for females with POI.
Thank you so much, Dr. Pruchi, for giving such an excellent talk on relevance of genetic testing in primary ovarian insufficiency. My sincere thanks to Dr. Sunita, the chairperson, the chairman of the MSR, and Dr. Amit Patwardhan, our close friend, Dr. Sadhana, for giving me this opportunity to be chairing this session. Thank you.
Okay, so excellent talks and excellent presentations. And we have few questions here. There are a lot of compliments pouring in. Excellent webinar and talks. The first question is, it is open to all the faculties. Gene polymorphism can be checked in lab, and is it available in India?
Yes, it is.
Can I answer that?
Yes, Deepak, you can
yes,
take
I'm sure Pruchi is here and even Manjeet is here. Yes, there are enough labs who are checking gene polymorphisms. The unfortunate part today is that we don't have defined panels which can be very effectively used. But yes, many of the labs can develop this test for you, or there are some who are already offering it. So you just need to check it out. And yes, the FSH receptor polymorphism tests are being done in some of the laboratories.
Can I ask you one thing, Deepak, from your friend from Illinois? See, what I felt is it's a fantastic idea to have the IT department coming in and helping. That's what artificial intelligence is going to do with us. Only thing is, everything is with the follicular size and the FSH. It means the patient has to undergo one cycle to use their module to individualize.
Yes, so no, this is where they don't have to go to full cycles. On the second day of the hormone injection, when you start the IVF cycle, on second day you have to just specify what amount of dose and how many follicles up to what size have grown, and the model can predict you how much of FSH to give to get the best response. So every
that's
cycle
wonderful.
you can individualize.
That's wonderful. But I do believe that there will be a latent phase. That is wonderful, because in the same cycle, because the cohort will vary in different cycle, and there may not be able to predict precisely. But usually on day 2, day 3 of a stimulation, it may be still in a latent phase, and some cohort might have to come. Also, I felt suddenly on day 5, day 6, if you drastically decrease the dose, as they have shown in one of the case, you usually land up in poor quality oocytes.
That was what even my presumption was, and that was my first reaction, that no, we wouldn't get good quality oocytes, so we may not even get enough response. But she's tried around 50 patients up to now, and she says that that really works. But what she wants is now that I told her that you need to do randomized control trials and even good clean case control studies, or even the same patient with optimized doses and see it. So it needs more studies. My idea here was that we have to now start making use of these artificial intelligence, and this will be the way tomorrow.
Deepak, can I ask you a question for in the same line here? So it's very interesting, and I reckon your friend has done it only on one patient, right?
So she's done 25 patients up to now.
Okay. So, I mean, it would make sense in terms of, because if you're overstimulating with FSH, you're supersaturating the system, and that will stop the feed forward loop. And you wouldn't get a good response if you have more FSH in your system. That's one thing. So that completely makes sense what she said, what she's found. But my question would be, if there is a patient that has got a poor response and has got a mutation in FSHR or in the report or in the promoter, no matter what you do, they're not going to respond to it. So it has to be a combination of, exactly, it has to be a combination of AI and genetics that has to work together.
Absolutely. Genetics comes first and one has to screen it up. So I had a question, complementary question for you as well, that in women who have POI and genetic mutations, do you think that the male child which are born from them, if they're the carriers of the same mutations, would be infertile?
Depending on the gene. Depending on what your gene you're talking about. So the examples I gave you, for example, SOX8 or NR5A1, there is a good chance that the child might have infertility or even DST. On the other hand, the child could be completely normal and still carry that mutation. That's the biggest problem, that we have a lot of genetic data coming in. We are finding a lot of causes, but then with that, we are finding a lot of variability. NR5A1 is the biggest conundrum because there are so many genetic modifiers that are working within the same family. The same mutation can give different phenotypes. You cannot even say that if you have mutation X, it's limited only to this phenotype. No, that's a very, very complicated question. Like that is the most complex question you could ask me.
Yeah, Anu, there is a very good compliment pouring for you from Dr. Firuza Parekh. Hello, Anu, you need to give a 1-hour talk. She says,
Thank
I
you.
visit Srinagar and run an infertility clinic there. We see a very high incidence of POF and premature ovarian insufficiency in these women. They also have a high incidence of ovarian antibodies and antithyroid antibodies. Which are the genes that could do this? Any thought on this?
So in my very first slide, I said there are autoimmune causes and there are pediatric causes and there are metabolic causes, and they could be completely independent of genetics. It is just that maybe they have got an autoimmune disorder that is producing antithyroid antibodies, which may or may not have a genetic cause. So it has to be done at case per case basis. So I cannot just give you a blanket answer that, okay, if your patient comes with this, that's what you should look for. You can't do that because it could be completely independent of a genetic mutation. They might be reacting to a medical drug they have taken or a medical procedure they have had. They could be reacting to an environmental what's the word of it for it? Contaminant that they have been exposed to. They could have been reacting to something that has been in their vicinity. Because thyroid hormone, you can actually, without having a mutation, you can produce antithyroid body if you are antibodies if you are exposed to, let's say, higher degrees of radiations. So you'd be attacking your thyroid. That's the problem. So I can say, okay, look for TSH, look for T3, look for T4, look for genes, the TSH receptors, but that might not be the case for your patient. You might have to look at the patient history and figure out what exactly is causing that's for the patient with autoimmune disorder or antithyroid antibodies. And what was the second part of the question? Sorry.
I mean, she wants to know, with this high incidence of ovarian antibodies and antithyroid.
What do you mean by ovarian antibodies?
Anti-ovarian antibodies, she says.
What would be an anti-ovarian antibody? Sorry, I'm interrupting you.
Which are the genes that could be responsible for this is her question.
So I explained for the antithyroid antibodies, but anti-ovarian antibodies, I don't understand what she means by that. Maybe she can write to me and say what it is, and then maybe I can be more clear to her.
Okay.
I think what is equally important is today's webinar highlighted that you have a premature ovarian failure the regenerative medicine is coming up very fast. Maybe your autologous bone marrow derived or a mesenchymal cells, fat cells, or even in that matter, the PRP. One need to be very clear that first find out, is there any genetic problem? And that's why premature ovarian failure, before you can even advise these patients.
So PRP, I agree with you. It is at a reasonably advanced stage, and there are lots of animal experimentation going on. In some cases, they are working on the patients as well, where the clinical trials are going on. As far as the regenerative medicine in terms of stem cells and for therapeutic use, we are doing a lot of stem cell work, but that is still in a stage of trying to understand how the process happens. We don't even understand how normal process happens at this point of time. So everything we and others are extrapolating are from the animal models. And at the end of the day, the more NGS is teaching us is that we are not mice. So, we need to.
I still will say last 7 to 8 years, we have advanced quite a lot in stem cell therapy, and quite many babies now, more than 100 babies are there across. But what is most important word what you used is still a research, because we still don't know how these stem cells work, how they forget only in an ovarian, but in regeneration of parenchymal cells or pancreatic cells, and the way it works into the lung tissue, it's phenomenal. It's phenomenal. It do work. In neurological conditions like paraplegia and all, we do see amazing result, but we don't know exactly how. Yes. So more and more scientists pouring in the work with the human. In India, still it is under research, as you say, in European countries. So I think, Amit, you would like to take ahead?
Yeah.
There is one last question.
Okay,
Amit.
take the last question, please.
Yeah, this is from Shilpi Sharma. She says, great presentation. And is the 29G
minus 29GA changing a
transcription factor binding site?
Yes, it binds to a very crucial transcription factor, which is one of the key regulators of FSH receptor transcription. So yes, it's not allowing the transcription factor to sit correctly, and therefore the protein is not RNA, and therefore the protein is not adequately formed.
I think, Deepak, you simplified it so beautifully.
Thank you. Thank you.
Really.
Thank you so
much. You made it.
And I think we had 3 fantastic lectures, stimulating, and in the background of the Nobel Prizes getting announced this week, I see the potential in all my 3 speakers. Fantastic. I think fantastic basic research, and Deepak has put it so well. He has translated it into clinical modalities, and Prochi has taken us through her journey of genetics. So I would really like to thank, on behalf of MSR, Dr. Sunita, myself, and all our committee members for the fantastic 3 speakers and the 3 chairpersons. And my compliments go to Sadhana for having thought of such a wonderful topic. I think it not only involves genetics, but it also involves clinical practice, artificial intelligence, and I think regenerative medicine. So hats off, Sadhana. I think you were the cherry on top of the full series of webinars that we did.
Thank you.
So viewers, we take a break now from our webinars, but block the date for 20th December when MSR will come up with a full day conference, parallel 2 halls, along with your registration and a selfie point, so that we will make it as realistic or as virtually realistic we can do. Have a good weekend, a safe weekend. Look after yourself and your family. And thank you very much. Thank you very, very much to all my listeners. Thank you so much.
Anu, thank you so much. It is maybe still the working time for you, and we could see the sun and all. Thank you so much. Thank you so much for taking out time from your work hours. Thank
Thank
you.
you. Thanks for
Anu,
giving
Bye.
us this wonderful
Bye,
opportunity,
Amit.
MSR.
We'll
Thank
catch
you
up
so much.
more. Thank you.
Bye.
Bye.
Thank you,
Bye,
everyone.
everybody.
Thank you,
Thank you,
Prajit.
everyone.
Thank you, Deepak.
Thank you, ma'am. Bye-bye.
Thank
Bye-bye.
you.
Thanks,
Bye.
Prochi.
Bye.
Bye.
Anu, there is a message for you. Dr. Firuza will share her work on autoimmunity, ovarian autoimmunity with you. She'll be sharing her papers with you so that you know you can.
Absolutely. You can send her my email and
Yeah,
then.
definitely.
Thank you.
Thank you, Anu. Thank you, Dr. Prochi. Thank you, Dr.
Thank
Deepak.
you.
Thank you,
Thank you.
Patwardhan. Thank you, other chairperson.
Wonderful, wonderful webinar.
Thank you.
Thank you. Bye-bye.
Bye-bye.
Bye.
Bye.
Okay. Bye.
Bye.